Okay, hi guys, it's Jubilee again, and this is going to be our final review for the final exam. I'm going to, since this is normally supposed to be a 2-hour review, I'm going to split this up into 2 parts. The first part, we're going to be going over the Jeopardy review. It's posted on eCampus. I've made a couple changes to it just for this video, so those changes will not be posted in the version on eCampus, and that's incentive for you guys to actually watch this video, okay? And then the second video that I'm going to post is going to be going over some pictures for you guys. You'll hopefully see them as very similar, if not identical, to the ones that you will see on your exam. It's just not going to have the questions attached to them, right? It's going to be just like the midterm review, where we'll go over some of the things that the arrows are pointing out, and you'll just kind of have to try to learn from them and imagine what a question on those pictures would be like, okay? So going over quickly the format of the exam. So it's going to cover quizzes 7 through 12, so everything after the midterm, right? So it's not going to be over everything we've learned this semester, just quizzes 7 through 12. But that being said, I do still expect you to know what a fibroblast is, right? We still expect you to know how epithelium works. We expect you to know how connective tissue works, stuff like that. But in terms of it being cumulative, it will not cover specific systems or anything like that. The questions won't be focused specifically on the systems that we covered in quizzes 1 through 6, okay? So yeah, and then the format of the exam, so it's going to be 60 questions. That's 5 more than for your midterm. Your midterm was 55, so this one's going to be 60 questions. You'll have 75 minutes to do it, okay? So that's a little more than a minute per question. It will be open note, unlike your midterm, so hopefully that'll make things a little easier for you guys. But because it is open note, we have made it to where there will be no backtracking, okay? So once you submit a question, you cannot go back to it. And the purpose of that is obviously to make sure that you're not just looking up all the answers, that you kind of know them a little bit before, right off the bat, that you kind of know what the question is talking about. You can obviously refer to your notes, but we want you to know something going into the exam. So that's why we've enabled that, because it is going to be open note. Just like your midterm, it's going to be taken out of 325, right? That's what it says in your syllabus. But there will be 385 points possible. Okay, so that's a 60-point cushion. So you can get those extra 60 points, but that's not an automatic 60 points, right? You don't just get those 60 points. You obviously have to get those questions right to get those extra points. But the possibility of earning those points is there for you guys, okay? And this is just the question breakdown of everything. So these are all the topics from quizzes 7 through 12 that will be covered on the exam. And it'll be just like the midterm, where it'll be pulled from a question bank of about 15 questions. So every single one of these has a question bank of questions that I have selected to be on your final. And you will, for example, for the eye, you'll get 5 of those 15 questions pulled. Okay, for endocrine, you'll get 6 of those 15 questions pulled. And that's randomized, just so that you guys don't all have the exact same questions. But yeah, okay, so that's going to be the question breakdown. If you guys want to take note of that, I tried to be nice in the breakdown of things. But yeah, it's pretty even. So yeah. Let me think. Oh, and then there will also be a general or a guaranteed question bank. So if you'll notice, this does not add up to 60. There will be a general question bank just like in your midterm, where there are 2 questions from every section in the question bank. And I want to say you'll get like 6 of them, or I don't know. If you add all those up, it'll be this minus 60. The rest of the questions will be from that guaranteed question bank. And that's just where I have some more general questions in there. They're a little bit easier, so it's supposed to be nice for you guys that I've put easier questions in there, and you obviously have a higher chance of getting them because it's pulled out of a smaller bank and you're getting more questions from that bank. Okay, so hopefully that makes sense for you guys. But yeah, and then just like for the midterm, I tried to be nice for you guys, and I picked honestly some of the easier questions. Okay, so I'm not picking too many, or in my opinion, I'm not picking any questions that cover nitty-gritty details. Most of them are going to be general overall concepts, things that I think it's important for you guys to have a handle on, to show that you've learned something in this course, right? So most of them are going to be the strict identification based questions, right? So there's a picture there, and I want you to identify those cells, or maybe understand some of the functions behind those cells as well. And then some of the other ones are going to be just word-based questions, right? Where it's mainly talking about the overall function of that system, right? So those slides in the beginning of the PowerPoints or in the beginning of my TA reviews where it talks about the overall function of these systems would be a good thing to look at, okay? So hopefully that clears that up. I hate that I don't have you guys to ask me questions because I don't know if I've covered everything. But yeah, hopefully that clears that up. So now we're going to go ahead and go over the Jeopardy review. I'm really hoping you guys are going to actually watch this and pay attention to it. I don't know how many of you guys are just going to skip over it and complete the extra credit assignment to get the extra credit points, but please try to pay attention to this because I really am trying to help you. I go over the exam questions before I made this PowerPoint. I always go over them again and update it. I always make it a little bit nicer and give you guys more details every semester that I do this review. So it really is tailored to the questions that could potentially be picked for you on your exam, okay? So they're tailored to the question banks of your exam, and I really am trying to help you guys, so please just try to pay attention. All the extra information that I give to you that's not on the slides, or the extra information that I've added it because I went over the exam again and remembered some things that you guys might really need to know, okay? So that's my big hint. Please try and pay attention. I don't mind if you fast forward through this and, make it 2 times speed or whatever, but I want you guys to pay attention, okay, if you want to do well. Okay, so we'll go ahead and get started. We'll just start from the beginning, endocrine and integument. Okay, so what types of sweat glands are these and what type of secretion do they utilize? Okay, so hopefully you've realized we're in the skin, right? This is the dermis, right? We've got a bunch of irregular connective tissue going on in here, dense irregular connective tissue with fibroblasts. This is a hair follicle, right? We've got our erector pili muscle. And so this sweat gland right here, that's really bubbly looking, and is emptying directly into the hair follicle, hopefully you guys can remember, is a sebaceous gland. Okay, so this is a sebaceous gland, and the type of secretion that it utilizes is going to be holocrine secretion. Okay, holocrine secretion. And remember, that's where the whole cell is secreted as the product, right? So the whole cell is secreted into this hair follicle, and that's the product. That's the secretion of these sebaceous sweat glands. Okay, and that's kind of what makes your hair oily and stuff like that, right? So it goes into the hair follicle. This one B right here, these are your normal sweat glands. So this is the one that's more in charge of thermoregulation and stuff like that, right? So these are going to be your exocrine sweat glands, okay? And these little hot pink things are going to be the sweat ducts. These are going to be the sweat glands right here, the lighter staining ones. So these are your exocrine sweat glands, and the type of secretion that they're going to utilize is going to be merocrine secretion. So that's the normal type of secretion where it the secretion undergoes exocytosis. Okay, So exocytosis, so that's your normal kind of secretion, merocrine secretion of your eccrine sweat glands. Okay, so here's that answer. And then here is a picture of all the sweat glands that we covered for you guys to go over. So there's our sebaceous sweat glands, holocrine secretion. Eccrine sweat glands are going to be your normal merocrine secretion. And then just remember that there's a third type of sweat gland that's more rare in humans. It's more common in dogs and cats and stuff like that, but that's going to be your apocrine sweat gland, right? And they're really easy to remember because they undergo apocrine secretion, right? Apocrine secretion, and that's where a part of the cell is the product. So that's where it takes some of the cell membrane with it. Okay, so just remember all of these and their secretion types. But yeah. Okay, what type of cell is this and what does it do? So we're in the skin again, right? This is our epidermis, this is our dermis to get you oriented. There's a little Meissner's corpuscle, right? That's responsible for light touch. And then this cell right here is what it's talking about. Okay, so you see how this is this kind of lighter staining cell that doesn't look like all the rest around here on this layer. Okay, so as we're going through this, please try to answer these before I answer them, because that's really what's going to help you in learning. Just knowing whether you guess wrong or right, it doesn't matter, but just the thought of guessing really helps you learn. Okay, so try and guess what that cell is. Hopefully you guess that it's a melanocyte. Okay, so this is going to be a melanocyte. So you see how it's different from all the cells down here. Hopefully you know that all the cells down here at the base of the epidermis is the stratum basal, right? So this is the stratum basal, and interspersed throughout the stratum basal are going to be these separate cells called melanocytes. Okay, and this is what they look at like. And notice how I have this picture right here that has Dr. Johnson's arrows already cropped into it, right? I didn't add those in. So that's probably a good hint whenever you see those on these reviews, that I took this directly from a question, right? But yeah, so melanocytes, and then talking about the function of them. Sorry, this slide's kind of weirdly formatted, but the function of melanocytes, right, is to synthesize melanin, right, to make melanin. Melanocytes make melanin, and then they inject it to these other cells of the stratum basal, right? So they disperse it to the other cells, the other keratinocytes, to try to protect them from UV radiation, right? So it's protecting them from the sun. So this is going to be an example of a melanocyte right here. You see how it looks kind of separate from all the other cells. Here's a melanocyte right here. You see how it's kind of divided off, right? That's probably another one over there. And then these are melanocytes as well. Okay, the kind of darker staining ones in here. So yeah, and you can see how they branch out and they're distributing their melanin to all of these cells, right? And again, that's to prevent mutations from happening in these cells, because these cells of the stratum basal are rapidly dividing, right? They're undergoing mitosis, and so if they get defects in their nucleus because they're constantly exposed to the sun, that's how we can get cancer, right? So that's why we have these melanocytes, to provide pigment to the skin, and that's what they're supposed to do. So they're providing it to the surrounding keratinocytes, right? All of these are keratinocytes, but mainly it's providing it to the ones towards the base. Okay, and notice how I've given you all of these examples of melanocytes, right? So that might mean that it's important for you to recognize a melanocyte in all of these different diagrams. Okay, that's my big hint right there. Oh, I'll go back to the board. I don't know. Okay, yeah. So what gland is this and what is it storing? And then also, what's this cell circled in white? Okay, so this hopefully you recognize from endocrine, this gland right here. This is going to be the thyroid gland. Okay, so this is going to be the thyroid gland, and hopefully you remember that the thyroid gland, oh, sorry, I'm being stupid. Okay, sorry, I was being distracted. Okay, sorry. But yeah, so this is the thyroid gland, and hopefully you remember that the thyroid gland has these follicles within it, right? So all of these are going to be follicles. And then the cells around them are going to be follicular cells, right? So these are all follicular cells around each follicle of the thyroid gland, and they're going to be producing this pink stuff that's being stored in the thyroid gland, and that is colloid. Okay, so this is colloid in here. And that's just one of the functions of the thyroid gland. It's important in metabolism, right? So colloid is just a precursor to these important proteins that are important for metabolism, okay? But the big thing we focus on in the endocrine system are going to be the cells in between the follicles, right? So these are follicular cells, so the cells in between the follicles, that's what this one is zoomed in right here with this white circle. These are going to be parafollicular cells, right? So these are going to be follicular cells surrounding the colloid, and these are going to be your parafollicular cells. Okay, hopefully you remember what those make, but that's going to be calcitonin. Okay, so calcitonin, that's made by those parafollicular cells. Let's see if I can go. Okay, but yeah, so colloid, that's what's stored in the follicles. That thing in the white circle was a parafollicular cell, and it's making calcitonin, okay? And I do need you guys to understand the function of calcitonin, and the way I kind of remember it is it tones down the calcium, okay? We've got too much calcium in the blood, it's going to tone it down. So calcitonin is going to reduce blood calcium levels, and it's going to do that by inhibiting osteoclasts. So hopefully you guys remember what osteoclasts look like, right? They're those multinucleate cells in the bone, and that's what's going to break down bone and release calcium, right? So if we're inhibiting these osteoclasts from breaking down bone and releasing calcium, that's going to reduce blood calcium, okay? So that's those parafollicular cells with calcitonin. Just a quick reminder, I need you guys to understand this too. Remember that PTH, parathyroid hormone, does the opposite of calcitonin, right? So they work against each other to kind of maintain equilibrium, right? So parathyroid hormone is going to be released when we have low calcium. So it's going to raise blood calcium levels, and it's going to do that by stimulating osteoclast production, right? So we're going to produce more of those osteoclasts that are going to break down bone. It's going to release more calcium, and so we're going to have more calcium in the blood. Okay, so just remember what those hormones do. Remember, it's made by the parafollicular cells that calcitonin is in the thyroid gland. Okay, so we're back to endocrine system. So what organ is this, and what are these regions called? Okay, so try to answer this on your own before I do, but hopefully you know that this is the pituitary gland. Okay, so this is the pituitary gland, and remember we've got these distinct 3 different regions in the pituitary gland. So we've got the pars distalis. This is the pars distalis. In between these 2 main parts is going to be the pars intermedia. And then out here is going to be the pars nervosa. Okay, pars nervosa. But yeah, so make sure you understand those, and also make sure you understand the types of cells and the types of hormones, right? So this diagram right here, have this ready to pull up or whatever whenever you take your exam. Just remember you've got your acetophils and your basophils in the pars distalis, right? And remember what each one is going to make, what each hormone is going to make. And then remember that in the pars nervosa, this oxytocin is stored in there. Okay, so it's made up here by the hypothalamus, and it is stored in the pars nervosa in those herring bodies. Okay, hopefully you remember that. So that's in the pars nervosa where they're stored. Okay, So define a portal system and give an endocrine example of a portal system. Okay, so we've covered different portal systems, right? Remember, we learned how the liver has a portal system, right? And we also learned about the pancreas having a portal system. We learned about the hypothalamus and the pituitary having a portal system. But basically, what a portal system is it's where you have two sets of capillary beds that are connected with each other by either an artery or a vein, right? Because we said normally blood goes through a capillary bed and then goes straight back to the heart. It is not normal for blood to go through a capillary bed, go through an artery or a vein, and then go through another capillary bed, right? So when we have a special thing like that, it's called a portal system, where we have two sets of capillary beds. And the whole point of that is to modify the blood in the first capillary bed, right? So we're going to drop something off or pick something up in the first capillary bed, and that's going to affect a change when it gets to the second capillary bed. Okay, so the whole point of that is instead of dropping this change off into the blood and it having to go all the way back to the heart and then being distributed all throughout the body, we're just dropping it off in this local area and it's being transported to another local area. Okay, so it alters blood composition locally, not systemically, locally, right? It's not going throughout the whole body again, it's just going to that secondary capillary bed. So it alters it in that one capillary bed in order to affect change in that secondary capillary bed. Okay, and then the example that I really want you guys to understand is in the pancreas. Remember, we have those pancreatic islet cells, so those islets of Langerhans in the pancreas, and the pancreatic acinar cells. Okay, so remember, those islets of Langerhans are going to produce insulin and glucagon, and they're going to put it in that first capillary bed, and then it's going to travel to the secondary capillary bed in these acinar cells to tell them, hey, we either need to start secreting pancreatic enzymes, because that's what these cells do, or we need to stop secreting pancreatic enzymes. Okay, so it's providing a local change between these two because they're so close together. So it's these guys sending these guys a message on what to do through insulin and glucagon. Okay. Not good at this. Okay, we'll go eye and digestive. So what are these two areas called? So this is the eye, hopefully you know that. And what liquids are found within them. Okay, so this first one right here, this is going to be in front of the lens. So this big area right here. So this is going to be the anterior chamber, right? Anterior meaning before or in front of. Okay, so this is the anterior chamber, and what it's going to contain is aqueous humor. Okay, aqueous humor. And that's going to be the more liquidy fluid, more liquidy liquid in there, I guess. So it's more runny in here, if that makes sense. It's not as thick as what is back here. So that's why it's the aqueous humor. It's more watery, if that helps you. Okay, and then back here, this is behind the lens. So this is going to be the posterior chamber, and it's going to contain vitreous humor. And so the liquid back here is really thick, okay? It's really cohesive, if that makes sense. So it likes to bead up and stay together. It's not as watery as the fluid up here. And that's needed to maintain the pressure and the shape of the eye, okay? That really thick liquid. Okay, so that's vitreous humor. So just remember that anterior chamber, posterior chamber of the eye. Okay, so we're back in the eye again, hopefully you realize. So these structures help maintain the spherical structure of the eye. What are they? Okay, and again, notice, hint, hint, we've got these arrows already in here. I didn't put those in here, so maybe I stole this directly from a question, right? And I'm trying to help you guys out. So let's see what we're pointing to. So A is pointing to this little hole in here. And to orient you guys, this is going to be the base of the iris branching out from the ciliary body. Okay. So this is the iris coming out right here. This is the anterior chamber. He gives you that, right? So this hole right here is going to be the canal of Schlemm. Okay, canal of Schlemm. This right here, the stuff blocking in front of the canal of Schlemm is going to be the trabecular meshwork. Okay, and then this right here, this is part of the ciliary body, right? And so these are going to be the secretory cells of the ciliary body. And so they're going to be secreting that aqueous humor, right? That aqueous humor, it's going to go all the way around the other side of the iris, and this trabecular meshwork is what's going to allow the aqueous humor through to the canal of Schlemm to drain, but it's not going to allow too much, and it's going to allow just enough, right, to maintain that pressure of the eye. So go over that again if you need to, but basically it's trying to keep the eye at the correct pressure, right? So we're constantly producing fluid over here from these secretory cells, okay, from these ciliary processes, and it constantly needs to be drained, but we don't want it to drain so fast that the eye deflates, and we don't want it to drain so slowly that the eye inflates, okay? So hopefully that makes sense, okay? So canal of Schlemm, trabecular meshwork, ciliary processes of the ciliary body. Okay, name the major functions of the digestive system. Okay, so like I said, there are a bunch of questions that are identifying things, and there are a bunch of them that are just functions of the system that we're talking about, right? So hopefully you can name the major functions, but I'll just give them to you. So secretion of digestive enzymes, absorption of nutrients and fluid, and transportation of food. Okay, and let's try and think of some examples of each of those organs in the digestive system in case they were to be in a question, right? So secretion of digestive enzymes, that's to help break down food, right? So some organs that are going to do that in your digestive system are going to be organs like the pancreas is going to secrete digestive enzymes. The stomach is going to secrete a bunch of digestive enzymes, right? Organs like that. Absorption of nutrients and fluids. So organs that are going to do a bunch of that are going to be your small intestine and your large intestine. They're going to absorb all of that nutrients that you've just digested from those digestive enzymes. And then transportation of food. When we think of transportation of food, the great example of that is going to be the esophagus, because that's all it does, right? Pretty much, is it just transports food from the oral cavity to the stomach. So a good example is going to be your esophagus. I'm bad at this, I'm sorry. Okay, so back to digestive. What are these 3 layers of the small intestine called? So this is the small intestine, right? We've got these villi in here. And so this kind of darker purple stuff lining the outside of the villi, remember that's going to be our epithelium, right? So this is the epithelium lining those villi of the small intestine. And this is going to be that simple columnar epithelium, right? And it's going to have that brush border on it. So ciliated, simple columnar epithelium. And it's going to have those goblet cells in it as well that are making mucus, right? Those goblet cells interspersed throughout here. This one right here, so this part is going to be the connective tissue under the epithelium. So this is going to be what's called the lamina propria, right? So this is that connective tissue inside the villi of the small intestine. So this is a bunch of connective tissue, and this is where we have a bunch of blood vessels and lymph vessels, right? Because we're absorbing food out here, and that needs to be picked up, processed by these cells, and put into the blood or the lymphatic system. So that's why we've got a bunch of blood vessels all in here. Okay, so this is the lamina propria with connective tissue. And then this one down here, way on the outside of the small intestine, this is going to be the muscularis externa, right? So this is what's important for peristalsis and contraction of the small intestine, right? So remember, we've got those two layers. We've got a longitudinal and a circular layer going in opposite directions of smooth muscle of the small intestine. And hopefully you guys remember what the name is for the nerves that are in between these two layers, right? The cell bodies of the nerves, that is Arbox plexus. Okay, you can't see it in here, but just know their location is always going to be in between these two layers of the muscularis externa. That's the Arbox plexus. Okay, but yeah, Meissner's plexus up here. But yeah, those are the layers: epithelium, lamina propria, muscularis externa. Okay, what are the layers of the retina? So this is the retina right here. This would be where the light's coming in. This would be where it's getting absorbed back here. So coming from here, so right here, this would be the ganglion layer. Okay, so this would be the ganglion layer. And these are the nerves that are, or the neurons that are eventually going to become the optic nerve, right? They're going to exit out through the eye, through the blind spot in the eye as the optic nerve. So this is the ganglion layer right here, and they're picking up that signal from these cells right here, and you can see all their nuclei lined up. These are going to be the bipolar cells, okay, bipolar cells, and that's because they're bipolar because they're touching these cells over here and they're touching these cells over here. So they have two poles and they're just transmitting this signal from here to here. Okay, so these are the bipolar cells, bipolar layer. And then down here, this whole layer right here, these is going to be this photosensitive portion of the retina. So these are going to be the rods and cones. Okay, and remember, the cones are going to be the thicker ones, right? They kind of look like cones, and the rods are going to be the more thin ones, right? Cones are for color, so they pick up colors and details. Rods are more for motion, so they just pick up light, dark, right? So changes in light, dark are perceived as motion, right? So you have more of them in your peripheral vision because it's more important to have motion sense to pick up changes in motion and stuff like that in your peripheral vision. You have more of these in your central vision, your cones, because it's more important to have detail in the center of where you're looking at, okay? And these are all the nuclei of the rods and cones, so they're just pushed up out of the way to the top so that these guys can do their part, okay? So photosensitive portion, and then this is just going to be the pigmented epithelium, right? Pigmented epithelium. And again, that's just to make sure when this light comes from the top and goes in, that it sinks into this black layer, this darker pigmented layer, right? Because dark things absorb light, right? Black absorbs light. So it's going to sink in here, absorb that light instead of bouncing right back and refracting, because then we get this kind of ping pong effect of going in and out and in and out, and we don't want light refracting like that and going back and forth, because that'll just confuse these cells whenever they get that second signal. So we just want light to go in and sink in right there. So that's that pigmented epithelium. And then down here under the retina, so this isn't part of the retina, but under it, where we have a bunch of blood vessels and stuff like that, that's going to be the choroid layer. So that's the vascular layer, supplying blood to all of these, okay? Because we don't have blood vessels in here, we have it in the choroid layer. Okay, so there you go. Okay, so this is the urinary system. And to be honest with you guys, I know you guys don't like the urinary system. It's mainly because I don't like it, so I probably don't teach it as well as I do the other systems. I'm not very good at it, to be honest. But so I tried to be nice with you guys when I chose the questions for the urinary system, okay? So that's my big hint, and I will tell you that there are not very many difficult questions for the urinary system. Okay, and so I've chosen very select pictures from the urinary system to show you, and that's basically telling you these are going to be the most difficult questions that you'll probably see in regards to the urinary system, okay? And it will likely be these exact pictures, right? Because I've already got those arrows there, those 3 arrows. So yeah, that's my big hint for you guys. Okay, so what are these? So we're going to start off with A. Hopefully A is the easiest for you guys. And this right here should be really helpful to you guys, okay? So this is showing you that we're in the medulla of the kidney. The medulla of the kidney, okay? And that's really important in understanding what structures are common in the medulla of the kidney, right? Because if you look at the structure of the nephron, there are only a certain amount of tubules that go down and dig deep into the medulla of the kidney, okay? So one of them is going to be this one right here. It's the same as this one right here and this one right here. So hopefully you guys have realized this is a collecting duct. This is a collecting duct, okay? The reason I know that is because the nuclei are very regularly spaced apart, okay? These cells are very big and thick, as well as really regular, okay? So it's a very thick, regular tubule. It's evenly spaced. So this is a collecting duct. This is a collecting duct. This is a collecting duct. There are tons of collecting ducts in the medulla of the kidney, right? Because that's where they're collecting all that urine and getting ready to go out of the kidney. So this is a collecting duct, okay? Another thing that you have in the medulla of the kidney is going to be the vasa recta. Vasa recta. So that's that name for those special capillaries in the medulla of the kidney, okay? So this is going to be the vasa recta right here. These are the vasa recta in the medulla of the kidney, okay? And then the other structure that makes up a lot of the medulla of the kidney is going to be the loop of Henle, right? Because remember we said that loop of Henle dips down into the medulla to create that concentration gradient, that high concentration gradient, so that the collecting duct can benefit from that. So these are the thin loops of Henle, okay? So just try and remember that. But yeah, so vasa recta, the paratubular capillaries. Yep. Okay, so this is more kidney stuff. What are all of these images depicting? Okay, so they're all depicting the same thing. I've already told you this is in the kidney, so this is a tubule in the kidney. Okay, it's not the highest resolution, but hopefully you can figure it out. So you see we've got all this fuzzy stuff on the inside of the tubule. There's fuzzy stuff on the inside of the tubule. This is an EM of that fuzzy stuff. Okay, super zoomed in. You can see that we've got these nuclei that are irregularly spaced throughout here. Okay, they're irregularly spaced. So when we have nuclei that are irregularly spaced, we know it's either the distal tubule or the proximal tubule, right? And if it has this fuzzy stuff in the center that this is showing right here, that's the brush border, right? And so which one has the brush border? Hopefully you know that's the proximal tubule, okay? So these are all examples of proximal tubules. That is my big hint. They're all examples of that. Okay. So now we're going to identify these. And so we'll start off with A. So here we are again in the medulla of the kidney, or closer to the medulla of the kidney down there. So A is pointing to these separate things. So these, hopefully you know, are going to be the ascending and descending loops of Henle. Okay, ascending and descending loops of Henle. And remember, we learned that the descending loop of Henle is very defined and the ascending loop of Henle is very bubbly, right? So that's how we know this is definitely ascending loop of Henle. Very bubbly, very defined. So this is the loop of Henle, okay? B, remember we said the blood vessels that are very common towards the medulla, once we get towards there, is going to be the vasa recta, okay? And here's this one really helps you because we've got a white blood cell in here, right? So we know this is a blood vessel. So vasa recta of those paratubular capillaries. And then in here, So we've got these tubules, they've got irregular nuclei spattered throughout, right? The nuclei are irregular. And if you look in the center, we do not have a brush border. Okay, in this stain, the brush border would be a much darker purple. So we do not have a brush border in the middle of these. So these would be the distal tubules. Okay, because we don't have a brush border. Hopefully that helps you guys. Okay, name the major difference in function between the gallbladder and the urinary bladder. So they're both bladders, right? They're both bladders, but obviously don't get them confused. Sometimes he likes to mix them up in answer choices, so don't let that trip you up. But they are both, since they are bladders, they're both responsible for storage of things, okay? The gallbladder is obviously going to store bile. The urinary bladder is obviously going to store urine, okay? But the main difference is that the urinary bladder does not concentrate that urine once it gets into the bladder, right? The organ that's responsible for concentrating urine is the kidneys, or are the kidneys, okay? So the urinary bladder just stores it. The gallbladder is going to store bile and concentrate it while it's in there, okay? So it stores and concentrates bile, urinary bladder does not, okay? Another big difference is the type of epithelium we have lining them, right? So remember, the urinary bladder has what kind of epithelium? Right? It has transitional epithelium, and that's to allow for it to increase in size without increasing the pressure within the urinary bladder, so that you don't burst when you are holding your urine, right? The gallbladder, instead, because it's concentrating that bile and it has to process that bile when it's in the gallbladder, has simple columnar epithelium, okay? So just remember those. Yep. Both store waste/fluids. What are the endocrine-like functions of the liver, and what are the exocrine functions of the liver? Okay, so remember, endocrine ones are going to be ones that end up in the blood and going to the rest of the body. Exocrine ones are going to be secreted outside of the body, and in terms of the liver, it's going to be secreted outside of the body into the digestive system. Okay, so that's considered an exocrine secretion of the liver, right? So the endocrine secretions are going to be glucose and plasma proteins, and those are going to go into vascular circulation. Okay, so they're going to go into the blood, glucose and plasma proteins. Exocrine secretions are going to be going into the digestive system, so that's going to be things like bile, right? So what's in bile is bilirubin and bile salts are the main constituents of bile, okay? And those are not going to go into the bloodstream, right? They're an exocrine secretion, not an endocrine secretion. So just keep that in mind. Okay, respiratory system. So identify. So hopefully you realize where we are. We are in the nasal cavity of the respiratory system. So we're going to identify these three things. So these big white kind of holes, I guess you could see here, these big white spaces within the nasal cavity are going to be the swell bodies. Okay, so all of these are big swell bodies. Hopefully you remember that, right? And remember, what those do is they swell up if you have some kind of reaction to whatever is going in the nose, right? So if there's a toxin or something like that your respiratory system really doesn't like, and we need to increase the filtration of the respiratory system, these little vessels are going to swell up with fluid, and it's going to close off the nasal cavity so that we're way increasing the amount of filtration that the air has to go through, right? And so that's also really important in allergic reactions, right? Because your body's freaking out and it doesn't know what's going on with all this pollen or whatever it's breathing in, so it swells up, right? When you get a cold, whenever you have allergies, that's what those swell bodies are doing in your nasal cavity, okay? B, what it's pointing to is this lining right here, that lining right here, and so that's going to be respiratory epithelium. right? Respiratory epithelium. So that's going to be that ciliated, pseudostratified, pseudostratified columnar epithelium. Sorry, that's your respiratory epithelium in here. And then see what it's pointing to. So these darker little lumps all in here, these darker lumps are going to be Bowman's glands. Bowman's glands, and remember those are going to be producing mucus secretions. Okay, so remember those, and then remember the other things that are in your respiratory system that we need to know, right? So there's olfactory epithelium. Make sure you know what that looks like. Make sure you know what the nerve bundles look like in here, right? Because we've got lots of nerve bundles under that olfactory epithelium. Make sure you remember that we do have bone and we do have cartilage, right, in the nasal cavity. And so make sure you know what those look like in the nasal cavity as well. Okay, those are my hints for that. Okay. So more on respiratory epithelium. What is this airway called? So this is an airway in the respiratory system. Hopefully you can see there's all this respiratory epithelium lining the inside, but the big hint on telling you what the name of this airway is are these right here. Okay, so try and guess what these are right here. Hopefully you recognize these are cartilage plates. These are cartilage plates, right? And so they're keeping this airway open. And so the way the airway goes, right, is it starts in the trachea. And remember, the trachea has nearly complete cartilage rings, right? Nearly complete cartilage rings around almost all of the trachea. And then we split into the bronchus. The bronchus has cartilage plates, so it's not around the whole thing, but we do have these plates of cartilage. Okay, and then those are going to split into the bronchioles, and the bronchioles no longer have cartilage. So if you don't see cartilage, it's not a bronchus, right? It's a bronchiole or something even smaller and later. Okay, so this is a bronchus. Note the cartilage plates. If you see cartilage plates, it's the bronchus. Okay, what are the differences between type 1 and type 2 pneumocytes? So remember, those are the cells in the alveoli of the lungs. And hopefully you remember that type 1 are going to be very flat and thin, like the number 1 is. So they're very flat and thin, and if we want a flat and thin cell in the lung, you'd think it would be so that gas can exchange through it, right? Because it's very flat and thin, and it's easy for gas to exchange through those very flat and thin cells. So that's going to be our type 1 pneumocytes. They're going to be very flat and thin, and they are primarily there for gas exchange, okay? Gas exchange, not air exchange. That's my big hint. We exchange gases, not air, in our lungs. Type 2 pneumocytes are going to be fatter, like the number 2, okay? And they're fatter because they're secreting things, right? They're making secretory products. And what is the big product that we learned about in the lungs that is very important is going to be surfactant, right? So these type 2 pneumocytes are producing surfactant. And remember, what that's going to do is reduce the surface tension in the lungs so that it's easier to inhale. Okay, it doesn't take a lot of force to inhale and inflate the lungs. Okay, right there. So we've got our flat type 1 pneumocytes for gas exchange, our bubbly type 2 that are making surfactant. Okay, what portion of the respiratory systems help with inspiration and with expiration? Okay, so this one's pretty straightforward, but just take the time to understand it. So inspiration is always an active process. And so the muscle that is primarily responsible for that is the diaphragm. Okay, but then the other muscle that kind of helps out that we need you guys to know is going to be the external intercostal muscles. Okay, so inspiration is external intercostal muscles. Expiration is going to be the natural elasticity of the lungs, because remember, that's a passive process, a passive process. They just passively collapse because the lungs are elastic. They want to collapse because they don't like being stretched out by active inspiration. So you just relax and they'll naturally collapse in. So the natural elasticity of the lungs. But if you need to exhale even faster than the time it takes to just relax, you have your internal intercostal muscles for that. Okay, so yeah, just remember that, and remember that these are kind of flipped, if that helps you remember, right? So inspiration, external intercostal muscles; expiration, internal intercostal muscles, right? So the E and the I and the E and the I. Okay, hopefully that helps. Okay, how does mucus in the conducting portion of the respiratory system function? Right, so mucus has a lot of functions in the respiratory system, so make sure you know them. So it's going to detoxify those gases. It's going to wash away current chemicals so you can smell new ones, right? It's constantly washing away things and dust and whatever. And it also contains antibodies to prevent infection, okay? And notice the things I've underlined because they're the things that are easily changed, right? So if I said mucus in the respiratory system toxifies gases, you would say no, it detoxifies gases. If I said it washes current chemicals or keeps current chemicals there so you can smell new things, you would say no, it washes away those chemicals. It doesn't keep them there. If I were to say it contains antibodies to create infection, you would say no, it prevents infection, right? Another thing he likes to do is change out antibodies for antibiotics, but the things I've underlined, really pay attention to, okay? Don't let these questions trick you on those tiny little things. Okay, male and female repro. What are some similarities and differences between the gonads of the male and female reproduction? Okay, so we talked about this. Remember, in the male, the main goal is to just constantly make sperm, right? Constantly make sperm at a steady rate. It doesn't matter what's going on, what time of the month it is, we are going to make the same amount of sperm today as we did yesterday, okay? The females, as well as testosterone, right? That doesn't really change much. In the females, we have this whole cyclic thing, right? So we're still producing the female gamete. So we're still producing ovum like the male are producing spermatozoa, but it's in a cyclic fashion, right? A cyclic and periodic fashion. So in the female, we are cyclically secreting hormones and periodically releasing ovum. And remember which ones those match up to, okay? So remember, it's cyclic for hormones and periodic for ovum. Okay, if I switched those, if I said periodic hormone secretion, that would be wrong. Okay, if I said cyclic ovum release, that would be wrong. Okay, so yeah, so that's the big difference, right? Males are always doing the same thing. Females have this whole cycle going on. Both, I forgot this part, both are controlled by FSH and LH. Okay, and remember, those are made in the pituitary gland. FSH, follicle stimulating hormone, is made in the pituitary gland to stimulate those follicles to mature. And LH, luteinizing hormone, is made in the pituitary gland, okay? And those help with hormone secretion in the male and the female. And in the female, they obviously help with the whole cycle thing going on as well, okay? But yeah, I noticed in a lot of you guys' review submissions, you said luteinizing hormone was made by the corpus luteum, which is false. That is false. Luteinizing hormone is made by the pituitary gland. Remember, we learned that in endocrine. It's made by the pituitary gland, and it has an effect on the ovaries and forms the corpus luteum. That's why they have the same name. Not because it's made by the corpus luteum. Remember, the corpus luteum makes progesterone and estrogen, but the reason it turns into the corpus luteum is because of this luteinizing hormone having an effect on it. Okay, so go over that again. Make sure you understand that. But yeah, both are responsible for secondary sex characteristics as well. Both of the gonads, right? So what is this and what does it empty into? So I'll give you a hint. This is in the male. This is a duct system in the male reproductive system. Okay, and if you look, we've got this scalloped appearance, right? So not all the cells are the same height as the others, and we've got these little cilia on here, okay? And these are true cilia on here, okay? So hopefully you realize this is an efferent duct in the male testes, okay? Efferent duct coming from the reedy testes to the epididymis. So that's what it empties into. It empties into the epididymis. Okay, so those are efferent ducts. They empty into the epididymis. And remember, the epididymis is the site of maturation and storage of sperm. Okay, really important. Ooh. Okay, so what are these? So I'll tell you, this is in the female reproductive system. These are in the ovaries. Hopefully you can recognize that. Again, notice I've got these lovely arrows that I kept here for you guys. So we're going to identify these. So A, what is this right here? So this is a follicle, right? We need to figure out what stage this follicle's at. But you can see we've got this oocyte in the middle, and you see how these cells around it are starting to get bigger and thicker, right? So this would be a primary follicle. This would be the oocyte within there. B is pointing to these cells around the oocyte in both of these follicles. So this is a primordial, this is a primary follicle, but it's still pointing to the cells around it in both of these follicles. So I'm going to say B is pointing to follicular cells of the follicles. Okay. C, what is this pointing to? So here's our germinal epithelium, right? Our germinal epithelium. And down here, right under it, this is connective tissue, right? These are fibroblasts. This is connective tissue anchoring this epithelium down to all the tissue under it, right? So we're going to call this connective tissue, and we're going to call it the tunica albuginea, okay? Tunica albuginea. And remember, that's the name. It basically means white coat, right? The tunic is a coat. Albuginea, that's like white, like albino. So it's that white coat of connective tissue. Okay, what creates the blood-testis barrier? So that's really important for you guys to understand. So we're in the seminiferous tubules of the male, right? So this whole thing is the seminiferous tubule. Right out here, we've got some Leydig cells. And so what creates the blood-testis barrier, hopefully you know, are these Sertoli cells, right? Sertoli cells. So these are the Sertoli cells. They're really easy to see in this stain because you can see these little granules within them, those nurse cells. So these are all Sertoli cells at around this level. So here's the best example of a Sertoli cell. And you can see it branching in between all the cells around it, connecting to the other Sertoli cells. That creates that blood-testis barrier. And remember, what it's protecting are these cells on the inside of it, right? Because remember, we said that these cells are new to the host because they're undergoing meiosis, right? They weren't there at birth like they were in the female. Okay, they started being created at puberty or whatever. So the immune system, these blood vessels out here where the blood is not used to these new cells, right? We're undergoing meiosis. We're changing the genetic makeup of these cells, right? That's the definition of meiosis. That's the whole point, right? To make a different offspring. So if the blood came in contact with these, it would not recognize these cells as itself. It would recognize them as foreign and attack them, and the male would never be able to produce any sperm because they'd always be dying, right? And so these Sertoli cells are creating this blood-testis barrier to protect these spermatocytes and these spermatids while they're undergoing meiosis from being attacked by the blood, okay? By the blood. This spermatogonia right here on the base does not need to be protected by the Sertoli cells because remember, they only undergo mitosis at this point. Okay, so if you need to review that, please do, but just make sure you understand everything that's going on, right? We have mitosis here, Sertoli cells, these are nurse cells, they also make the blood-testis barrier, and then we've got spermatocytes and spermatids undergoing meiosis and developing into little bullet-like late spermatids in here, okay? We've got these Leydig cells, remember I already pointed out, these are producing testosterone. These are those myoid cells on the outside of the seminiferous tubules. We've got some fibroblasts, some blood vessels. Okay, just pointing out some things that you might need to know. But yeah, okay. So that's important. Last one. So this is female repro. This is the cervix. So what are these? And again, I have these lovely arrows already pointed out on here. So A, we're going to say A, this is in the cervix, and it's even telling you we've got simple columnar epithelium in this area, right? So this is going to be the endocervix, So that's in the cervical canal in here, right? Where we are not supposed to have contact with the outside environment. We're trying to make everything sterile, and we're trying to create a favorable microenvironment for fertilization to occur. So that's the endocervix in here, where we have simple columnar epithelium. B, right here, so this is pointing to the junction where out here we have stratified squamous epithelium, right? And in here we have that simple columnar of the endocervix. Okay, so this junction between these two types of epithelium, remember, was the ectocervix. This is where we're changing the type of epithelium, right? And then remember this right here. So this is like the hole but in the cervix, the opening of the cervix. That's going to be the external cervical os. External cervical os. Okay. If I were to swap this word out for internal, it would be wrong. Okay. Hopefully that helps you guys. I'm giving you all a lot of hints, so please, I hope you're watching this. But okay, so that was the end of this portion of the review. If you have any questions, try to email your TA or message me. I will try to be responsive as I can be. But yeah, please pay attention to all the things I pointed out in this review. Tell your other classmates to actually watch it, because I really am trying to be helpful for you guys. And this really is supposed to be a good review for your test. Okay. And yeah, and then I will now make the second part of the review where we will be going over pictures.